So our first step looking at the diversity of life is to look at viruses. And you might be thinking, but viruses aren't alive. You're correct. So a virus is an infectious particle, and it's basically made up of two main parts. It's got an outer protein covering, and then inside it's got some DNA or RNA, some genes. And viruses are not alive, right? Viruses are not made up of cells. They cannot carry out their own metabolism. They have to have some kind of host cell. They cannot reproduce on their own. They have to have some kind of host cell to help reproduce. But what they can do is they can evolve. So they can mutate their DNA, they can respond to stimuli, and they can evolve using natural selection. So that's why we are going to look at them under the diversity of life, because viruses affect life. So viruses were first discovered in 1800s, the early 1900s. And you don't have to remember these names or dates, but this is kind of a story for you. The first virus isolated was the tobacco mosaic virus, and this was on the tobacco plant. And what was happening is the tobacco plant will get a sick and disease when it has the virus. Like you can see, these leaves here are not looking well. At first, this was thought to be a bacterial pathogen. And the first step in figuring out this virus, this was transmissible, is Mayer took some sap from a sick plant and then rubbed it on a healthy plant, and they figured out that the sap could transmit the virus, which they thought was a bacterium. So to try to isolate the bacterium, in 1893, what they did was they filtered it. So they filtered the sap through this filter that should have prevented bacteria from passing through. So ideally, if the bacteria, because bacteria are much larger than a virus, so if the bacteria got filtered, they would be up here and they would remain up here. And then this would be the filtered sap that should be clean of bacteria. So then when they rubbed that on a healthy plant, that plant became sick. And at this point we knew, okay, well, maybe it's some kind of bacterial toxin or something that the bacteria are secreting into the sap instead of a virus. They didn't know what a virus was. And then eventually we tried to grow this infectious agent on media, and they could not do it. So they could not take this infection sap and grow it on like a petri dish. They had to grow inside of this host plant, which is weird because if it was a bacteria you would be able to grow on a petri dish. And it wasn't until 1935 that using an electron microscope, we were actually able to crystallize the virus particle and see it. So we now know this as the tobacco mosaic virus, and this was the first virus we were able to isolate. So viruses are made up of two main structures. They have some kind of nucleic acid, meaning they have RNA or DNA. And then they have an outside protein coat called a capsid. This is made up of protein, and this is nucleic acids. Sometimes outside of their protein coat, they have another membrane layer. The membrane layer does not come from the virus itself, it comes from the host of the virus. And here are the four main different kind of structures of a virus. You can have these long tubular viruses like the tobacco mosaic virus, where you've got kind of this long protein coat, and then the RNA or DNA kind of makes a helix inside. This is the shape of things like the Ebola virus, those long tubular viruses. This is an adenovirus. So an adenovirus has these kind of geometric shapes where they've got these geometric shapes that form together to make this overall kind of enclosure called the capsid, and then inside you can see the DNA. These protein spikes on the outside, and you can see these here, are going to help that virus recognize the cells that it can invade. This is an influenza virus, so this is a circular virus. Inside, we've got RNA and protein in a complex. And then outside of that, they've got this membrane. And this is a phospholipid bilayer membrane, but it's not produced by the virus. What happens is when the virus bursts out of its host cell, it grabs a little piece of membrane from the host cell and takes it with it. So this is an influenza. This is also going to be very similar to the structure of the coronavirus. And then there's another set of viruses here. And this one looks a little like an alien. This one's called a bacteriophage. And these are the most well-studied viruses because these are the viruses that invade bacteria. So bacterio meaning bacteria, and then phage, like to eat. So these are viruses that infect only bacterial cells. And they have a little bit of a more complicated structure. They have this kind of protein head with some DNA in here. And then they have this long tubular tail area with these tail fibers. And what happens is they land on the bacteria with these little spikes down here. And then they inject the DNA from through the tube into the bacterial cell. Viral genomes are varied. They can be made up of DNA or RNA. Some viruses are really, really small, having only 3 genes. Some viruses are really large, having 2,000 and more genes. So if you're made of DNA or RNA, the viruses can also be linear or circular, meaning that they have straight chromosomes or circular chromosomes. And then, depending on their linear or circular, they can be single-stranded or double-stranded. The only option that's not available is a double-stranded RNA, if that's circular. That doesn't happen. But you can see there's a huge variety in the different types of DNA that can be found in viruses. The coronavirus, COVID-19, is a RNA virus, and it has about 2,600 genes inside of it. So if we look at this outer protein structure, it's called a capsid, and it encloses the virus on the outside. So the capsid are made up of little sections called capsomeres. So the virus will have DNA that codes for these little proteins, capsomeres. And what happens is the virus sends this DNA to the host cell. And then the host cell will actually produce these capsomeres for the virus. And then virus assembly is really cool. It actually just happens spontaneously without any energy input. So the host cells don't really build the virus. But what can happen if you took all of the parts needed to make a virus and put them in a cup and shook them up, they would self-assemble into the correct shape of the virus. So that's what I mean by spontaneous. The least energy using shape for a virus is the shape that they're in. And so over time, these viruses have selected these shapes that they just build themselves. They don't even need worker bees to build them. Some viruses have a viral envelope. And this is, again, not built by the virus, but it's built by the host. So what happens when the virus bursts out of its host cell, it takes a piece of the host cell with it. And it spends time. The DNA for the virus codes for these protein coats. And so the host cell makes these proteins, puts them in its own membrane, and then when the virus leaves, it takes a piece of this membrane with it. And so the viral envelope actually contains viral components, like the proteins, and host cell molecules, like the phospholipids in the membrane envelope. Over here, we've got an influenza cell, and these have that kind of capsid, that envelope on it on the outside. And then this is the novel coronavirus that causes COVID-19. And so you can see the structure inside here. We've got capsid proteins and RNA inside here. And then all of this part out here is the envelope, and that's all derived from the host cell. So the actual COVID-19 genes code for these spike proteins. The host cell manufactures these spike proteins, sticks them in its own envelope, and then when this virus bursts out of the cell, it takes this envelope piece with it. Typically, viruses that have these envelopes tend to be more virulent, causing diseases in animals. Most of the time, these envelopes are going to help the virus infect its host cell. We'll give a little bit of attention to bacteriophages. So normally these are the viruses that get the most attention because we've studied them the most. Right now, coronaviruses are going to get a little bit more of our attention. But these have kind of this interesting elongated shape. And so you can see down here is a bacterial cell, and then this is the bacteriophage virus. And it kind of lands on it like a spaceship. And then the spikes in the bottom help anchor the virus to the cell. And then the DNA is actually inserted through this tube into the plasma membrane of the cell, where then it can infect the cell and go on to continue to replicate.